is a study of a cell. Cytology is a study of a cell. Obviously, there's a lot of cells in the body. We have 1 trillion cells in the body, and we actually have 250 million different types of cells. We do not have to know all of them. But these are different kinds of cells. There's muscle cells, there's nerve cells, there's red blood cells, there are reproductive cells, and there's a number of cells. But this is the general schema for what a cell looks like internally in a multicellular organism. Since we're taking the human anatomy and physiology, we're going to deal with multicellular organisms. This is what a cell looks like with all the stuff inside of it in a multicellular organism. The major structures of the cell are the plasma membrane, which is the barrier around the cell. And it's a very specific barrier. If we didn't have that barrier, then anything could go in and out of the cell that wanted to go in and out of the cell. And before long, the cell would rupture and die or swell up and die, okay? We have a nucleus, which is right here. This big old structure. It's the biggest structure, biggest individual structure inside the cell. It contains all the genetic material for the entire organism. So every cell in our body, whether it be a big toe cell or a scalp cell, contains all the DNA, okay, information for every part of the body. And then finally, we have the cytoplasm, which is the gel-like substance in between the plasma membrane, not including the nucleus. So everything outside the nucleus, but inside the plasma membrane, and that contains organelles, which are these defined structures, cytosol, which is the watery gel-like fluid inside the cytoplasm, and inclusions. If you think of an inclusion as a chlorophyll or water inclusion in a plant cell, or some other type of inclusion, a glycogen granule inclusion, they are just storage depots. We don't have very many inclusion bodies in human cells. The plasma membrane is a individualized structure. When we studied lipids last time, the first one we studied was triglycerides. Okay, triglycerides was a glycerol backbone with 3 long-chain fatty acids. We then discussed phospholipids. This is what the cell membrane is made up of: a bilipid layer of phospholipids. The outer part, this is outside the cell, this is inside the cell. The outer part is the phosphate group. Remember, a phospholipid consists of glycerol backbone with 2 long-chain fatty acids in the phosphate group. The phosphate group faces both outside and inside. The phosphate group is polar, therefore it is hydrophilic. The lipid groups are nonpolar. They are hydrophobic. It is fortunate that these groups are hydrophilic because all the material, all the fluids that are bringing materials to the cell, the majority of that, as we learned last time, is water, okay, and it is polar. And if we did not have a polar molecule here, we would not attract the water molecule, which contains the glucose, the oxygen, all the other stuff, okay? Same thing on the inside of the cell. The inside of the cell is primarily water, okay? And so we want to be able to exchange waste products to the outside of the cell, but because this is water in here, we need to be able to get close to the plasma membrane so we can undergo exchange of materials here. And so the plasma membrane is a bilipid layer, biphospholipid layer, okay? It's also a fluid mosaic, which means it's not static, okay? If you were to see a schematic movie, an animated movie about the plasma membrane, it would look like it was flowing back and forth. It has certain points of attachment, but it looked like it was flowing back and forth. And that's why they mean by, so it looked almost like a wave-like structure. The phospholipid bilayer, basic fabric of the membrane, it is semi-permeable. What that means, it will let some things in, but it won't let everything in. Or it will let some things in certain concentrations based on the needs, okay? It is semi-permeable. If it wasn't semi-permeable, if it was freely permeable to water, at least 75% of the body is water. It was freely permeable water. It is the stuff inside the cell, we'll call that particles or solute, is very concentrated. If this membrane was freely permeable water, water would go right through the membrane and keep coming into the cell, until either equilibrium was reached, which means the concentration of water inside the cell would be equal to the concentration of water outside the cell, which is highly unlikely, or until the cell would rupture, okay? So that's why it is selectively permeable. It's selectively permeable to water and a number of other things. Now, O2 and CO2, they can go right through it. Obviously, we want O2 to go right through and CO2 to go right through. Lollipop-shaped phospholipid molecules that are polar. Hydrophilic water-loving heads, that's the heads that point outward. They are attracted to water in the intercellular inside the cell, extracellular fluid. Extracellular fluid is also called interstitial fluid, okay? Intercellular fluid and extracellular fluid. Extracellular fluid is also called interstitial fluid. And we have extracellular fluid, but we also have fluid inside the vascular space, okay? All cells are surrounded by capillaries. We'll learn that when we get to chapter 17, Anatomy and Physiology 2. We just went over the blood chapter not too long ago in Anatomy and Physiology 2. The hydrophobic portion of the lipids are these fatty acid tails, the two fatty acid tails, which limit the amount of polar substances coming through the membrane. We also have lipids which are attached to sugar groups. Sugar groups are important, sugar groups. So if we have a lipid here, and this is a sugar group up here, this is a sugar group up here. We have lipids which are attached to sugar groups. And I can't get this to go up and down if I'm screen down like that. We also have cholesterol. These yellow things right in here, they are cholesterol molecules. They stabilize the phospholipid membrane, so they stabilize membrane and decrease fluidity, okay? You want to have a wave-like action here in the fluid mosaic model of the membrane, but you also want it to be stable enough to not break away from the cell and to be attached to the inner workings of the cell, because a lot of these proteins here, if you look at this, one of these integral proteins actually has portals here for things to pass through, okay? And so we want to have some fluidity to be able to move the membrane around, but we also want to have some stability, and cholesterol is one of the things that stabilizes it. The carbohydrate-rich area, so any of these attachments out here that have carbohydrates, they are responsible for cell-to-cell recognition. One cell would like to recognize the other cell, and that's one of the things that carbohydrates do. Integral proteins are these proteins that are either go all the way through, they're transmembrane, or they're embedded on one side, okay? Or like I say, it extends all the way through. Peripheral proteins are these proteins out here that are loosely attached to the peripheral. Peripheral proteins are loosely attached to the integral proteins. Membrane proteins are responsible most of the specialized functions of the membrane, which is primarily transport, okay? Specialized functions: transport, receptors, attachment to the cytoskeleton, okay? So attachment to the cytoskeleton here internally, and attachment to the extracellular matrix, because a lot of these cells, when we get to the tissue chapter, which is chapter number 4, the slides and the Pearson assignments for chapter 4 are already open. If over the weekend you want to get ahead, you don't have to, but they're already open. Attachment to cytoskeleton, enzymatic activity, intercellular joining. We want one cell to be able to join next to the cell, and cell-to-cell recognition. Okay, how does things get transported through the membrane? Things must move back and forth from the extracellular to intercellular space. These are the 4 processes that are passive transport. That means they don't require energy. Your lab for this is PhysioX 10.0. Make sure you follow the instructions for this lab explicitly, because you need to get to the PhysioX 10.0, not the PhysioX 9.1, because that requires Adobe Flash Player. And most computers won't work with Adobe Flash Player anymore because of the security concerns. That's why PhysioX 10.0, and it's explicit in the instructions as to how to get there to get to the two labs. There's two labs, and what they are simulated labs. One of them's going to be on osmosis and osmotic pressure, which is the movement of water, okay? And the second one's going to be on active transport, which are two of the most important ways that things move through the membrane. So we've got these passive methods of moving through the membrane. I look at passive as a car being on top of the hill, and once you give it a slight push, you know, it will go down to the bottom of the hill. So diffusion, everybody knows about diffusion. That one person comes into the room and opens up a can of sardines. This happened in medical school. Roger Simpson, one of my friends, he used to like to eat sardines. So he would bring sardines if we had a conference or something, lunch conference. And once he opened those up, you could smell it throughout the entire lecture hall. It's the same way with some kinds of cologne and perfume. Once somebody comes in, you can smell it throughout the entire lecture hall. And what all diffusion is the movement of particles from an area of higher concentration to an area of lower concentration, down a concentration gradient, of course, because you're going from higher to low, until equilibrium is reached. So if we're talking about Roger Simpson's can of sardines and the odor from that, okay, the odor from that, at some point when equilibrium was reached, if we could sample portions of the air in the front and the back of the lecture hall, assuming all the doors were closed and all the air was circulating uniformly, we'd have the same number of sardine particles. The same way with the cologne. After it had disseminated throughout the entire lecture hall, or the entire room that you're in, okay, we could at some point sample and it would be the same number of particles in every place, because this is just a matter of moving throughout a solution. You can see it, you've all seen it before. You take a pitcher of water and you dump in Kool-Aid, okay? If you allow it to stand, the Kool-Aid will disseminate throughout the entire solution. What we do is take a stir and make it go faster, but it will disseminate throughout the entire solution. That is diffusion, just moving down a concentration gradient through no barriers, doesn't have to have a barrier, anything like that.
Professor.
Yes?
Is it different from osmosis?
Osmosis, yes. Osmosis is the movement of water.
Okay, thank you.
Okay. And we haven't gotten to that. We're going to get to that pretty soon, okay? Diffusion, particles of solid move in area high concentration to area low concentration. It can be through a membrane, but doesn't have to be. We talked about diffusion in a room. We talked about diffusion in Kool-Aid, okay? But it can be through this membrane, okay? Diffusion can move down through this membrane, does not require energy, okay? So if you've got a fat-soluble molecule that's not too big, remember, things that go through this membrane have to be fairly small. Otherwise, they have to be helped through the membrane. If you have a fat-soluble particle that's going to go through, that's transported here by some kind of carrier, because it's not going to mix well in water. So when we get to learning about fats, and that's probably in anatomy and physiology too, we'll find out that fats transported in blood are transported by carriers, okay? But they move from an area of high concentration to area of low concentration, okay? Facilitated diffusion, still no energy required. So this is one of those portals here, okay? This is one of those portals that we saw that went all the way through the membrane, okay? Whatever needs to go inside, okay? And so this is a sugar molecule or amino acid. Some sugars can be very long. We talked about glycogen, some of the sugars can be very small, monosaccharides. This thing opens up like maybe a trunk, okay? Well, not a trunk, something that's got an entrance and exit to it. So this thing opens up based on some kind of stimulus, and this particle, the sugar molecule or this amino acid molecule, comes in here. It then undergoes a configurational change. Okay, and we're talking about one of these integral proteins. It undergoes a configuration change, and now the extracellular part, the part that was facing out the cell, closes, and the intercellular part opens up. And now this is big molecule, which was too big to get through this membrane by itself, is able to get through this membrane, still going down a concentration gradient, okay? But we have this concept called facilitated diffusion. It means that this structure here has helped the molecule get through by undergoing a configuration change. So it has essentially become a channel. So this is facilitated diffusion. Osmosis is the diffusion of water and water only. Osmosis is the diffusion of water and water only. Nothing else osmosis, okay? Osmosis is the movement of water. The scientific definition of osmosis, osmosis is the movement of water from an area of high concentration of water through a semi-permeable membrane to a lower concentration of water, down a concentration gradient until equilibrium is reached, if it's possible. Scientific definition of osmosis is water moves down a concentration gradient from an area of high concentration to an area of low concentration through a semi-permeable membrane until equilibrium is reached. You will also hear the definition, water moved from an area of low concentration, that's low solute concentration, that's low dissolved particles, to an area of high concentration, okay, that is a high solute concentration or a larger number of dissolved particles. And you know that means that if you put a cell which has an area of high concentration, solute concentration, number of particles obviously inside the cell is much greater than the number of particles outside the cell. Then if we didn't have a semi-permeable membrane, if we just had a membrane, the water will go through that membrane, and the cell will swell up. The water will go through that membrane and the cell will swell up. So osmosis scientifically is the movement of water from an area of high concentration to an area of lower concentration of water down a concentration gradient. In other words, it's trying to dilute whatever it's moving into, because whatever it's moving into is more concentrated than what it's leaving from. So you can think of either water in high concentration going into the area where water is in lower concentration, or you can think of water which is in a solution which has a low number of solute particles is moving to a solution which has a high number of solute particles, and it's attempting to dilute it's not doing it on purpose, but it's going to dilute the number of solute particles. They are specialized channels which water moves in the cell membrane. They're called aquaporins. Those are channels for water movement. Okay, this is an example of osmosis with a freely permeable membrane. Freely permeable membrane. In other words, water can go back and forth as much as it wants to. So we've got this half of the tube. Now the solute particles can't go through the membrane, but water can. We've got this many particles on this side of the tube. I take that back. The solid particles can move too. Or osmolarity, granular osmosis. What's going to happen is, because it's freely permeable, the water is going to move here to dilute the number of particles, and the particles are going to move here to increase the number of particles on this side. So if we have increased water concentration here and decreased water concentration, notice the levels are the same. So that means we have more particles dissolved in water on this side than we do on this side. Solute is going to move from this side to that side because it goes down a concentration gradient. Water is going to move from this side to that side because it goes down a concentration gradient. At some point, it will reach equilibrium where it has the same number of solid particles, same number of red particles, and the same amount of water because they're equal. That is if you have a freely permeable membrane that everything can go, that the elements that you're looking for through can go through. Now, if you have a selectively permeable membrane, okay, which means it's selectively permeable to water, selectively permeable to water, we have then water is going to go from an area of low concentration of solid particles to an area of high concentration of solid particles. Water is going to go from an area of low concentration of solid particles to an area of high concentration of solid particles. Or you can think about it like this. Water is going to go from an area of higher concentration of water, because both of these chambers before the movement, before they've pulled the cover off this membrane, are at the same level. But because there's fewer solid particles and they're at the same level, there must be more solution. You know, the level of a solution is based on the things that are dissolved in it and the amount of liquid. Well, there must be more water here than there is here before this membrane. So water is going to go in this direction from an area of high concentration of water to an area of low concentration of water until, and through a semi-permeable membrane, down a concentration gradient until equilibrium is reached. Till equilibrium is reached. So this is the state that's equilibrium. So we have 5 particles here of solid. We haven't changed the number of particles of solid, but we have decreased the amount of water and added to this side. And if we were measuring the amount of water and the amount of solid particles per milliliter, they would probably be equal. They would be equal because equilibrium is reached. But just remember, osmosis deals with the movement of water through a semi-permeable membrane down an area of higher concentration of water to a lower area concentration of water until equilibrium is reached. After you do the lab, which is be due in 2 weeks, after you do that lab, you will understand osmosis a whole lot more. Okay, you'll understand it a whole lot more. And this is just an illustration of what we talked about. Osmolarity. Osmolarity is the concentration of solutes, the solid particles in the blood. Osmolarity is the concentration of solutes, which are dissolved particles in the blood. Tonicity, okay, is how many solutes are in the solution. Less solutes in the solution, something is hypotonic, okay? We take a red blood cell, we place it in a hypotonic solution, okay? Normally, a normal isotonic solution, if we're talking about sodium chloride, is 0.9% sodium chloride. On the wards, you call that normal saline, okay? I'm going to hang a bottle of normal saline, or if I don't need all the salt, I'm going to hang a bottle of half normal saline and a quarter normal saline. But normal saline is isotonic, which means it has the same number of particles outside the cell as it does inside the cell. So let's say that we have put some red blood cells in a hypotonic solution. In other words, there is less solute here, there's less solute here. And so water is going to go into the cell, water is going to go into the red blood cell and cause the cell to swell. And eventually it could cause the cell to rupture. So if a red blood cell is placed in a hypotonic solution, which has less solutes in solution, then the water will go from outside. Remember, it has less solutes, it has more water. So water is going to go out from outside, from the area of less solutes inside the cell to an area of more solutes. If we have the opposite, if we place a cell in a hypertonic solution, so if we place the cell not in a 0.9% solution, which is isotonic, the same as the cell, say we place the cell in a 5 normal solution. Now we've got more water inside the cell than we do outside the cell. We've got more water inside the cell than we do outside the cell. So the water is going to come out of the cell and cause the cell to crenate. And this is crenation, okay? This is called crenation. And so remember that term, you might see that term, crenation. That occurs when you have a hypertonic solution, which means the number of particles in the solution is greater than the number of particles inside the cell.
It's
and
like
shrinking? Yes, go ahead.
Is it like shrinking?
Yeah, crenation shrinking. That's what it is. See, it went from this nice biconcave sphere to this sort of ball with these little spikes on it. So yeah, that's what it did. It shrank. Okay. It'd be the same thing if you I can't think of an example now, but that's what it is shrinking. Yeah, crenation and shrinking are the same thing.
Thank
An
you.
isotonic solution. This is isotonic. No net water, no net gain. Now, water's going to go back and forth, okay? We have equilibrium. That's equilibrium. That means things are moving back and forth, but there's no net gain. So we put these red blood cells in a 0.9% normal saline solution. You don't have to remember that. It's called normal saline, okay? 0.9% sodium chloride solution is called normal saline. And they will still stay the same shape because the number of particles outside the cell is equal to the number of particles inside the cell. Water concentration inside the cell is equal to water concentration outside the cell. So they won't swell, they'll stay the same size. So those are the three things. That is hypotonic. If you put a red blood cell in a hypotonic solution, the red blood cell is going to swell up. If you put a red blood cell in a hypertonic solution, red blood cell will shrivel. And if you put an isotonic solution, there will be no net change in the red blood cell. Water's going to go in and out, but there's going to be no positive or no negative net change, okay? It's not enough to know the definitions. You need to be able to explain this if you got a short answer question. And copying and pasting from the slides is going to get you a 0. Copying and pasting from the slides are using the exact words of the slide. In other words, it will say explain placing a red blood cell in a hypertonic, hypotonic, and isotonic solution, what happens to the red blood cell and why. So you can look at that and explain that in your own words, okay? I'm giving you examples, word to the wise. If I say I'm giving you an example, you're probably going to see something like that, okay? That would be a short answer question. Remember, at least 25% of your test is going to be short answer questions. Short answer questions are not up on the internet. You can't Google the answers because I make them up as I go. They come from primarily from your notes, from your student notes, and from the slides. General rule for solving tonicity problems, water goes where the stuff is, okay? Water goes where the stuff is. If there's a lot of stuff outside the cell, water's going to go outside the cell. If there's a lot of stuff inside the cell, water's going to go inside the cell. If the stuff inside and outside the cell are the same, water is going to have no net gain. So you can expect a tonicity problem. I used to give it as extra credit when we took exams in person, okay? Filtration. Imagine a pressure head, okay? Imagine a pressure head inside somewhere with little, imagine a pressure head in your hose and there's little pinholes in the hose as you go out. That could be a sprinkler hose, okay? That could be a hose that you lay down in your garden or in your lawn and you put water going through it and the little holes cause the water to go out in all different directions over your lawn, okay? This is called filtration, okay? This is called filtration. And it is a process that happens in multiple places in the body, but especially in the kidneys, okay? What happens is a pressure gradient builds up inside the kidney unit of the kidney called the glomerulus, okay? And most of the fluid inside that vessel actually goes out into this glomerulus, okay? So because of the filtration pressure, most of the fluid goes out, and some glucose and some other things, the big protein molecules can't go out because these holes are small. And fortunately, in the kidney, all of it, 99.9% of it gets resorbed. You know, about 1 liter, we have about 1.5 liters of urine, but the rest of it gets back absorbed. Otherwise, when passed through the kidney, we'd lose all our blood volume, okay? But the blood pressure pushes this fluid, okay, pushes this fluid, and because of the pressure and because of the leaky membranes in the kidney, leaky membranes in the glomerulus, it actually goes out because of the pressure, okay? And that's what filtration is. That's another way of passively moving. There's no energy involved, okay? We just have a pressure head, a natural pressure head. The way we have a pressure head is the arteries that come out are smaller than the arteries that go in. So if you've got bigger arteries that go in and the arteries that come out smaller, then you're going to build up pressure just because of that. If your outflow, if the entrance into the theater has got 4 cross doors, okay, you can get a lot of people into the theater, but if your exits down the front have one person at a time, it's harder to get that group out. And you can have a lot of pressure buildup. Well, that's what you do in the glomerulus and kidney. And if you have leaky membranes, and that fluid's going to come out and be collected, it's going to be, like I say, it's going to be reabsorbed. It's not a selective thing. The only selective thing about it is the big molecules don't come out because they can't get out the holes. Okay. The other part of your homework assignment is going to be on active transport. All the other things we talked about, diffusion, facilitated diffusion, osmosis, filtration, none of those require energy. They're all going down a concentration gradient. And filtration is a pressure gradient, but they're all going down a concentration gradient. And I compare that to that car sitting on top of the hill. All you do is take the parking brake out and it goes down to the hill and stops once it loses its speed, once equilibrium is reached. Active transport, and you'll notice that when I give lectures, I try to have visualizations to myself, because 3 or 4 or 5 years from now, when you're sitting there taking your boards exams and they throw this question at you out of the blue, okay, hopefully if you can remember that picture, you'll know what I'm talking about. Active transport is you and 4 or 5 or 6 of your buddies pushing that car back up the hill. It requires energy. And as a general rule, active transport is pushing something, as a general, not all the time, but as a general, pushing something gets a concentration gradient. So if we had a high concentration of potassium on one side and low concentration of potassium on the other side, potassium will come to the low concentration. Well, if we want to get that potassium back inside the cell or inside the nerve or wherever we want to get it going to, and we're going to go get some concentration gradient, it's going to take some energy. That energy is in the form of ATP, or if you got my visualization picture, energy is in the form of you and 5 of your classmates pushing that car back up the hill. You better hope it's a small car, okay? Energy is required in primary active transport, in secondary active transport, and in vesicular transport, okay? Primary active transport, how you do that, you phosphorylate a transport protein. That gives it the energy. The protein changes configuration. The solute is pumped into the cell, and this involves, we'll learn about the sodium potassium pump in the nervous system chapter in chapter 11, okay? This involves the sodium potassium pump at a number of places, and we're trying to pump one of these electrolytes against the concentration gradient. So we're priming up a transport protein, okay? We're priming up a transport protein, giving it some energy so it can move one of these electrolytes against the concentration gradient. So it can be pumped into the cell. And this is just an example of it, okay? Secondary active transport is powered by a concentration gradient. This is your buddy tagging along, okay? This is your buddy tagging along. Somebody says, hey, let's go to the drive-in. If you've ever been to a drive-in, it's $20 a car, okay? I'll pay the $20. And so your buddy tags along. It's powered by a concentration gradient, okay? As one molecule comes down its concentration gradient, another different molecule comes along. It can be a molecule that's actually coming down a concentration gradient, or it can be a molecule that has the opposite charge, okay? Has the opposite charge. In this particular instance, okay, we're pushing sodium against the concentration gradient. We're pushing sodium against the concentration gradient, and glucose comes along for the ride. So we're pushing one molecule, and we're pushing one molecule against the concentration, and then the molecule comes along for the ride. It's not important if it's sodium or if it's glucose. It's just one other molecule comes along for the ride, going down the concentration gradient or coming along because charge. You will see that a lot in some of the GI transports and red blood cell transports. When you move a positive ion, and chloride will follow it because it has a negative charge, okay? Lastly is vesicular transport. How do we get really big molecules into a cell, okay? Or how do we get garbage, really big garbage out of the cell. We can get very small lipid solid molecules through the membrane just freely. O2, CO2, some of the small fat soluble molecules. The bigger molecules, we got to use one of those transport proteins, whether it be through facilitated diffusion, whether it be an aquaphor for water, or whether it be active transport. Okay. In this particular case, we're talking about something even bigger than that. We're talking about a big molecule or a bacteria or some foreign material that we've got to take inside the cell. Okay. And so we have processes called endocytosis, where endocytosis means we're going to take it inside, we're going to engulf it. Okay. We're going to engulf it. So we're going to take something inside, and we'll say this is a bacteria. We're going to take it inside. We're going to attach it to a lysosome, which we'll talk about a little bit, and that's going to break it down. Okay. And then we're going to use whatever we can use and put the rest of it out. We have penocytosis, which is a very small pinching in. Okay. It's for things that we can bring in, usually fluids of some kind, something in a fluid thing. You know, we already have special things for water, but we may have some other fluids we want to bring in. Okay. And we can just, instead of engulfing, you know, instead of just taking a huge portion of the membrane and engulfing around it, we can take a very smaller portion of the membrane because this fluid that we want to bring in is so much smaller. We can do phagocytosis, which is a specialized part of endocytosis, where we're bringing in a foreign particle. Okay, we're going to bring in a foreign particle, and we're going to isolate that particle. We may try to build antibodies against it. We may try to break it down into its antigen parts. Okay, that's usually what you try to do. The cell does is bring in the foreign particle, break it down into its antigen parts. Another part of the cell takes its antigen up there onto the surface of the cell, and then exposes it to white blood cells to see if you can either cellular response or an antibody response. That it will be in anatomy and physiology too also. Okay. So that's another thing. We can take in some material, usually foreign material, might be foreign bacteria, might be if you had a tiny piece of metal or splinter or something like that, because things get walled off in the body. If you ever go after a piece of foreign material, most of the time it's walled off. Okay. And if it's walled off, it's not infected, then there's really no sense, unless it's next to a vital organ, unless it's, you know, impinged on the spinal cord, there's no reason to go after it. You know, it's laughable. It's laughable on TV and movies and stuff to watch people say, I've got to get the bullet out right now, or you're going to die from infection. The bullet's traveling at a high rate of speed when it enters your body. Okay. It's very hot when it enters your body. There's no reason to have to go get the bullet right away. And the other thing is, they always close every wound. Okay. Well, you never want to close an infected wound. So those are two laughable moments. Now, when you watch movies on TV, that they've got to go get the bullet. I've got to get the bullet out or you're going to die. Well, that's an entire falsehood. If it's not bothering anything, it's not critical that you go get it right that minute. Okay. Exocytosis, we use a number of things. Okay. The thyroid gland is an endocrine gland that produces thyroid hormone. It's an endocrine gland. It doesn't have any ducts to duct anything into any other chamber, right directly into the blood vessel. So the thyroid gland produces the thyroid hormone. It is packaged up. It is packaged up and then taken to the cell membrane. And then the package, okay, the vesicle fuses with the cell membrane and opens up and dumps the thyroid hormone into the interstitial fluid. Well, like I told you a little while ago, every cell is surrounded by blood vessels. Okay, that's how the cell gets oxygen. That's how the cell gets and glucose and the other nutrients it needs and the electrolytes it needs. That's how it gets rid of waste. Okay, so we think of a cell as a flat one-dimensional thing when it's actually more of a spherical shape and it's surrounded by a capillary bed. And so this thyroid hormone is dumped into the interstitial space. The space is very narrow because the cell is surrounded by capillaries right almost adjacent to it. Okay. And so that thyroid hormone goes into those blood vessels and is taken to the body wherever it needs to be released. Same way with neurotransmitters, okay? So exocytosis is the opposite of endocytosis. It can be waste material that's ejected too, okay? Then we can have receptor-mediated endocytosis if a cell needs a particular thing, needs a particular, whatever the substance is, has a receptor for it, okay? Here's the substance, here's a receptor, its interaction with the receptor, and then that substance can be brought into the cell. Now we're going to, so that's transport. There is diffusion, facilitated diffusion, osmosis, filtration, active transport, and then ways to bring in the cell by either bringing it in or pushing it out. And some of them require mediators, some require receptors for it. Okay, but that's how we get stuff in and out of the cell. Okay, we're going to talk about these individual organelles in the cell: mitochondria, ribosomes, endoplasmic reticulum, lysosomes, peroxisomes, cytoskeleton, centrioles. Then we're going to talk about the nucleus and then inclusion bodies. Okay, this is the first one we're going to talk about. This is the mitochondria. It's called the powerhouse of the cell. This is where ATP gets produced. We are using right now billions of ATP. You and I are using billions of ATP right now. And so this is a mitochondria. It has a double layer, an outer membrane and an inner membrane that's folded in on itself. And this is called cristae, okay? The mitochondria is the only organelle that I know of that has its own DNA and RNA. Mitochondria is the only organelle that I know of that has its own DNA and RNA. And so at some point in our development, okay, if we talk about atoms going to molecules, going to organelles, going to cells, going to tissue, things like this, this may have been a structure that had the capability of living on its own because it had its own method of replicating itself, okay? Obviously, since that point, it's lost that ability, okay? Since that point, it's lost that ability. But this is the powerhouse of the cell. This is very important. Muscles have a lot of mitochondria, okay? The heart has the most mitochondrial per gram of tissue as anything in the body. The heart, because remember, the heart's beating an average of 72 beats a minute, every second of every minute of every hour of every day until you die, okay? And so we need a lot of energy there. So this is the structure that provides energy, and energy is in the form of ATP, okay? Double membrane organelle, oval shaped, intermembrane, highly folded. It's called cristae to increase the surface area. That's where most of the reactions that produce the ATP occur, okay? Outer membrane is smooth and featureless, contains their own DNA and RNA to reproduce themselves. Powerhouse of the cell, site of cellular respiration, okay? Remember, plants take in CO2 and water in the presence of energy, which is sunlight, okay? And make glucose and oxygen. Oxygen is actually a byproduct of that, which is good for us, okay? We then take in that glucose, whether we take it in as starch, whether we take it in as glycogen, whatever form, or just simple sugar. We take in that glucose, convert it to glucose, take in that sugar, carbohydrate in whatever form, convert it to glucose, and then we take glucose in the presence of oxygen. So the CO2 plus water in the presence of sun is called photosynthesis. Glucose plus oxygen is called cellular respiration. We then form CO2, H2O, and ATP. That's how we get our energy when we break down glucose, okay? When we break down glucose. So do not ever in your life be on an Atkins or any type of glucose sparing diet. In other words, where you're not taking in a lot of glucose. When you take any type of major exam, you're killing yourself. The brain runs on glucose. Brain runs on glucose. If you, for whatever reason, trying to get those last 2 pounds down before you go to your reunion, okay, or on an Atkins diet, Atkins, you can eat all the protein and all the fat you want to, you just can eat glucose, which is terrible for your heart arteries, okay? but it causes you to lose weight because you don't eat bread and all that other stuff. But you deprive yourself of glucose. We can burn other things, but we don't burn it efficiently. So if you've ever been to a bodybuilding contest and they tell the bodybuilders to turn right and turn left, yeah, about half of them will turn the wrong way because they're so glucose depleted. So we need glucose, and the reason we need glucose is that's how we make ATP. One of the other structures is a ribosome. Okay, we'll talk about ribosome in more detail. It's made of 2 subunits. Ribosomes function in protein synthesis. Okay, ribosomes are made. Okay, ribosomes are made in the nucleus in a specific structure called the nucleolus. They then go out in their subcomponents and through the nuclear pores. The nucleus has a double-layered nuclear membrane. They then go out through the pores of the nuclear membrane and go out either attached to endoplasmic reticulum. Okay. And we call that rough endoplasmic reticulum, or they remain unattached and go out into the cytoplasm, and we call those free ribosomes, and their endoplasmic reticulum, we call it smooth endoplasmic reticulum. So ribosomes are units, okay, we're going to talk about this in more detail. They're units where protein synthesis occurs, okay? If they're bound to the endoplasmic reticulum, that's ER. That's where proteins are made that are either going to be proteins in the cell membrane or be exported from the cell. If they go out into the cytoplasm or free ribosomes, that's where we're going to make proteins for use inside the cell. And this is rough endoplasmic reticulum. This is rough endoplasmic reticulum. These are little ribosomes attached to the rough endoplasmic reticulum. This is smooth endoplasmic reticulum, and I can imagine that this is probably a ribosome in the cytoplasm. So we have the nucleus here. Here's the nucleolus where the ribosomes are made. Ribosomes will then come out one of these nuclear pores, and they will either attach to the endoplasmic reticulum, which is really an extension of the nuclear membrane that's folded on itself. Or if they don't attach to this, they will go out into the cytoplasm. So you have 2 types of endoplasmic reticulum. Rough endoplasmic reticulum is only called rough because the ribosomes are attached to it. Or smooth endoplasmic reticulum, which has no ribosomes attached to this. I look at this as an assembly line. This is an outcropping of the nuclear membrane that's got multiple channels to it. And I look at this as an assembly line. And I look at these ribosomes as workers on the assembly line. And I look at this smooth endoplasmic reticulum as an assembly line. Well, it's automated and there's no workers there. Okay, so that's how I look at that. I look at in the cell, I look at pictures. I look at pictures in my head. All right, we're looking at endoplasmic reticulum. This is rough endoplasmic reticulum, smooth endoplasmic reticulum. This is the nuclear envelope. These are the ribosomes that you can see. They're attached to the endoplasmic reticulum. This is electron micrograph, okay, about smooth and rough. You would never have to identify, I would never give you a picture of this and ask you to identify that. I would even if we're having actual labs, I'd never give a picture and ask you to identify that. What is endoplasmic reticulum? There's structure interconnected tubes with cisterns, fluid-filled cavities, continues from the nuclear membrane. The rough has ribosomes attached. The smooth has no ribosomes attached. Rough is a site for protein production. Smooth is a site for lipid production, calcium storage in muscle cells, and we'll talk about endoplasmic reticulum, smooth plasmic reticulum when we talk about muscle physiology in chapter 9, okay? And detoxifies drugs in liver and synthesize steroids, okay? So you could get a multiple choice question says, which of the following is not functions of the smooth endoplasmic reticulum, okay? And so your exam is going to be time sensitive. You're not going to have all day long to do the exam, okay? It may be 2 hours, it may be 2.5 hours, but it'd be very time sensitive, so you'll need to know the material. Obviously, you have your notes. If you fill them in, you have your notes in front of you. You can print out the slides if you want to. You have your textbook or have your textbook open, but you're not going to have time to look up every individual answer. You're not going to have time to look up every individual answer. So you need to have a working knowledge and working familiarity of your notes, so that if you need to look something up, hopefully you don't need to look something up. Hopefully you remember that rough endoplasmic reticulum is protein production. And it's going to be proteins that's going to the plasma membrane to be exported outside the cell, okay? So that's endoplasmic reticulum. Remember, it's just an extension of the nuclear membrane. And the reason being is a signal that's going to tell us which protein to make is coming out of the nucleus and going to go to, so this is the central headquarters. We're sending a worker out, we're sending the fax, we're sending a worker out, a messenger out, down to the factory floor with the plans to build a protein. And the protein is going to start and be built in the rough endoplasmic reticulum. Golgi apparatus, okay? These are the next structures here. Imagine now we've got this protein built. We're going to go through here and we're going to go down these, okay? And we're going to modify that protein. Remember, proteins has 4 different types of structures. It has primary, secondary, tertiary, and quaternary, okay? We get a linear protein out of the rough endoplasmic reticulum. For a protein to be functional, it either has to be in the tertiary or in the quaternary structure if it's going to be attached. And it's going to have to be transported. Remember, if it's made in the rough endoplasmic reticulum, it's going to need to be modified, and it's not going to be transported. So the Golgi apparatus is this is my UPS store. You bring in whatever you built, hand it to them, they package it, they fold it, package it, get it ready for transport and put it on the truck. These are vesicles that are essentially breaking off of portions of the Golgi apparatus. This could be very well be thyroid hormones being produced. Thyroid hormone is the signal went out from the thyroid gland saying, hey, make more thyroid hormone. Something came into the thyroid, says got to make some more. Okay. The messenger brought the plans for the thyroid hormone out to the endoplasmic reticulum. The workers are sending the thyroid hormone. Okay. Golgi apparatus put it in this configuration, makes it functional, packaged it up in these little vesicles, and brought it to the plasma membrane to fuse with the plasma membrane so that it can go out into the interstitial fluid and be absorbed by the blood. Or if it's just a plasma membrane protein, we're going to bring it to the membrane and incorporate it to the membrane. Golgi apparatus is the Golgi apparatus. I look at that as the UPS store for packaging, and then as the UPS trucks or FedEx trucks, whichever one you want to talk about, as sending it out to wherever it's got to go. Structures, stacked membrane sacs that pinch off to form vesicles. Processing, packaging, transporting of products of the endoplasmic reticulum may also activate proteins if necessary. Golgi apparatus. Lysosomes. These are the garbage trucks, okay? They are made, okay? They are enzymes made made the typical way. Signal comes out from the nucleus, made in the endoplasmic reticulum, packaged up by the Golgi apparatus, but they stay inside the cell. These have some of the most powerful enzymes in the body, okay? If a piece of foreign material comes in, bacteria, viruses, toxins, that foreign material is engulfed. We saw endocytosis, which means there's a membrane placed around it. This lysosome comes and fuses. It's double membrane too. It fuses with the membrane of the endocytotic vesicle. It dumps its enzymes out into the vesicle and digests the particles. It can also digest old, worn out, or unneeded organelles. So if we got a mitochondria, it says, hey, I'm worn out. Lysosome will come over there, digest it. It will, the cell will reuse whatever it can, whatever amino acids and other things it can use. Whatever it can't use, it will be ejected out of the cell by way of exocytosis and probably picked up by the macrophages, by one of the scavenger cells in the body. Or if you need to break down glucose or storage racks of hormones, it can do that. That lysosome can do that. It also, when we're initially born, before we're born, we have webbing between our fingers. The lysosome is one of the things that do that. It can also break down bone if we need to have calcium done. Okay. We have peroxisomes. There's really small structures. They contain hydrogen peroxide. That's hydrogen peroxide right there. Notice the only hydrogen, water and hydrogen peroxide have the same number, have the same kind of atoms. They have hydrogen and oxygen atom. Water has 2 water molecules and 1 oxygen molecule. Hydrogen peroxide has 2 water molecules and 2 oxygen molecules. So same made up same elements, but obviously a different type of compound. It neutralizes free radicals, detoxes alcohol and drugs. Okay. So the peroxisomes, it's one of these little structures here. And if you know it makes hydrogen peroxide and takes care of free radicals and drugs. Next, we have a cytoskeleton. Cytoskeleton does a number of 2 things. Number 1, it shores up the structure of the cell, so the cell is not in constant, it's in motion, but it's not in constant motion. We've got to have the plasma membrane attached to the cytoplasm of the cell and certain organelles attached. So you can think of it as a little bit as scaffolding, okay? So rods in the cytoplasm give support to the cell and aid in movement. We have microfilaments. This is an actin microfilament. When we study muscle, we'll talk about the interaction between actin and myosin, okay? They brace the membrane and give it shape. They're involved in the motility of cell with myosin, so that's how the muscle cell contracts, the interaction between actin and myosin. And they're used for movement between endo and exocytosis. We have keratin, which are intermediate filaments, which provide more support to the cell, okay? Then we have microtubules that originate in the centrosome, and these are responsible for any of the cells that have cilia or flagella. The sperm has a flagella on it, okay? Any cells that either have cilia or flagella, those are the anchor points for that, okay? And then we have a centriole. Centriole provides support to the cell. It also provides tubules for movement of substance, okay? They're at right angles here, right angles to each other. And it also is where the spindle fibers come out when chromosomes are lining up before the cell divides in mitosis. The centrioles, microtubules organize an area, 9 triplets of microtubules, basal body for cilia and flagella, organized spindle apparatus paired at right angles. That's all if you know what's on this slide, obviously you know everything about it. We have cell extensions too, okay? These are microvilli. Microvilli, what they do, this could be part of the intestine. And so if you didn't have any microvilli, you'd have this cell surface right here that you wanted to absorb nutrients with. Well, if we add a whole bunch of these, we increase the surface area by a huge amount. And this gives us the ability to have more absorption area if we're talking about the lining of the GI system, okay? We also have cilia and flagella. Well, this is microvilli. We can have cilia and flagella. Cilia are little fan-like structures. Flagella, you know what we talked about, that the tail of the sperm, but cilia, we have cilia inside our respiratory tract that are fanning particles back towards our mouth. So little tiny particles that we ingest are caught by the mucus, and these cilia form a fan-like movement to try to get those, not the big particles. If you ingest a piece of popcorn, cilia is not going to move it. These little tiny particles are trying to move that back up into the larynx, to the pharynx, so that you'll swallow it, okay? Finally, the nucleus is the nucleus here. Nucleus has a membrane, okay? Has a nuclear envelope. It has chromatin, which is the DNA, which is surrounded by histone proteins, and the nucleolus, okay? Most cells only have one nucleus, except for muscle cells, and bone breakdown cells, and I think some liver cells. One per cell, except for skeletal muscle cells. When we get into tissues, which is chapter 4, we'll learn about different types of muscle cells. Some liver cells and bone destruction cells, which are the osteoblasts, which break bone down. Okay. So every cell has one, except for these three examples that have more. And then a mature red blood cell doesn't have any nucleus in it because it's ejected the nucleus. They are the control centers for every activity. Every piece of your body is contained in the nucleus of every cell in your body, the instructions for building that piece. It has signals that turn it on and off, okay? But every cell in your body is totipotent, totipotential, has the potential for becoming any other cell / organ in your body, okay? Large nucleus larger than other organelles, 3 distinct regions, the membrane, okay, double membrane, outside continuous endoplasmic reticulum, contains pores which things can go out. Nucleoli, dark staining, cytoribosome subunit, not membrane bound, maybe multiple in a nucleus. And then the nucleus also has the chromosomes, and the chromosomes are essentially DNA that is wrapped up with these histoproteins and continuously wrapped until you form a chromosome, okay? Inclusions are just small, they're storage chambers, okay? That's really all an inclusion is. It doesn't do anything other than store stuff. In a plant cell, you have water inclusions, okay? An inclusion body that stores water, because plants aren't like us. You know, we can go get water when we want water, okay? A plant can't do that. So you'll have a water inclusion, okay, that where the plant will store water if it needs to. You can have an inclusion for melanin, okay, melanin pigment. We'll learn when in chapter 5 in the epidermis and the skin that melanin pigment is stored in inclusions as it goes up. It's formed in the basal layers and it is then added to the cells and we get melanin pigment that's fill up, glycogen granules and lipids. It's also in a plant cell, it's where it stores chlorophyll. Okay, but inclusion's not per se a living thing, it's just a storage vessel, okay? And that's a cell. So let me get out of this. Let me turn everybody's mic, unmute all.
Dawson, for some reason, gets.
All right, that's not gonna work.
And we saw him on Tinder, and then Robin was like, invite him over and tell him to bring the goods.
Okay. Obviously, somebody's talking on the phone while we're lecturing, but that's okay. If you need to ask me a question, press your space bar and ask a question. Hold space bar and ask a question. Any questions on this part? Next one doesn't have nearly as many slides.
No, I'm good.
Okay, any other questions? All right, let me get the other slides up and we'll start on that. Cellular division. Close this now. I didn't close it. Cellular division.
You want to take maybe a 10-minute break?
Do I want to? No.
Okay.
You can take one. I'm recording the lecture. Cellular division. Okay. I haven't had any dinner yet. I don't get dinner till I get done. This probably will take 30 or 45 minutes. Cellular division. Why don't we just have one big cell, okay, instead of a whole bunch of multi-cells, okay? Why do we have just one big cell instead of a whole bunch of multi-cells? It has to do with volume of the cell and surface area. Everything that the cell gets has to go through the cell membrane. If you can imagine a huge, I can't imagine a basketball 10 times the size it normally is. A basketball 10 times the size it normally is. From that outside area, from the surface area to get to that center is a very long distance, okay? It would be prohibitive. So what would be happening if you needed water and you get waste in the very inner core of the cell, that just is not going to happen. That's just not going to happen. And because of that, that inner core of a huge cell would die, okay? And so it's a relationship of area to volume. Volume is the cube, okay? And area is the square. So as the volume increases by the cube, the area only increases by the square. So you get a bigger and bigger volume with a less proportional area. That's all you need to know. You don't need to know any equations. You certainly don't need to know that. It's really, why is the cell the size the cell is? The cell is the size the cell is because of the volume to surface area ratio. That's all you need to know. Life cycle of the cell. There's essentially two basic phases, an interphase and a mitotic phase. The mitotic phase is a division phase. The interphase is not a resting phase, okay? A lot of stuff is going on. Actually, the cell is growing, replicating the DNA, and then making the enzymes getting ready to divide, okay? So the interphase is not a resting phase. The always call it the resting phase. It's actually a whole lot of activity going on. But we have the interphase, okay, which is actually most of the cell, and then the mitotic phase, which is a much smaller part, which is where the division actually occurs. Resting phase from formation, cell vision, still metabolically active, also preparing for next division. We have the first is the G, carrying out cell function and growing. When a cell divides, when a cell undergoes mitosis and divides, it forms two identical daughter cells that are smaller than the original cell, because essentially you're dividing the cell in half. So if you take a cell and you duplicate the DNA and you divide the chromosomes so that each side gets 46 chromosomes, which is our number of chromosomes in a human, 23 pairs, 46 chromosomes, and you divide the cytoplasm in half. You don't replicate the cytoplasm, you just replicate the nuclear DNA. Then you got two smaller cells. Those cells have to grow, okay? So that's the first phase. That's the G phase, carrying out cell functions, because the cell's got to live, so it's going to have to include cell functions. Then we have the S phase, where we're now replicating the DNA. So once we divided the DNA, we had two daughter cells, with the same amount of cytoplasm, same amount of chromosomes, okay? And they're growing. That was the growth phase. Now we're preparing to divide again. Remember, stem cells divide all the time. Skin cells divide all the time because we have exfoliation, abrasion. I just put the dog the other day in the car and scraped my arm on the sharp side of the door handle and got an abrasion there and a laceration. Okay, and so those cells are replicating. So in the next phase, the first was the growth phase, the next is the S phase. We're carrying out our cell functions just like before. Growth phase, carrying out cell functions, growing. S phase, carrying out cell functions and replicating DNA. And then growth 2 phase, carrying out cell functions and synthesizing the enzymes and proteins necessary for division. So while we're in interphase, we're doing stuff, okay? We may be in the growth phase of interphase the most part. We may grow up to the size, and if the cell's not replicating very much, we may just stay in that growth phase where we're just carrying out cell functions, okay? But these are the three functions, these are the three phases of interphase, okay? So before a cell can divide, it has to replicate its DNA, okay? Before a cell can divide, it has to replicate its DNA. So each one of the 46 chromosomes has to replicate itself, because we're going to have two identical daughter cells. Each cell in the body has 46 chromosomes, 23 pairs. Well, we've got to replicate that. So the DNA unwinds, the DNA of a cell unwinds, and we're going to build one part of the cell based on these bases, A, T, C, and G. We're going to form a complementary strand of that. And on the other side, we're going to form a complementary strand to this portion of DNA. This is called semi-conservative replication, because on each one of these chromosomes now, we're replicating the chromosome. Say we're replicating in the male the Y chromosome, or in a male or female the X chromosome. Well, we've got a double helix. We're now replicating that chromosome. So we've got to unwind this DNA. We've got to unwind this DNA, and we've got to make an exact copy. Well, we've unwound it here. We've got an enzyme that unwinds it, okay? And we're going to make a copy by, if this is an adenine, it's going to be a thymine. If this is a cytosine, it's going to be a guanine, okay? We are reconstructing another exact copy of this DNA by using one of the original strands and using the other original strand and making 2 chromosomes now. So each strand is, yes,
Exact copy?
exact copy, yeah.
Okay.
See, because this strand right here, see the one we're making, this one we're making right here, this is the exact strand as this original strand right here. And this original strand right here, the one we're making here, is the exact copy of that because of the base pairs. You know, A can only bind to T and C can only bind to G. So we're making 2 exact copies, okay? 2 exact copies of the DNA, semi-conservative replication in the fact that we're using 1 original strand and 1 new strand that we made. And then, see it unwound here, we're going to build a strand and it's going to wind back again. Step 2, 2 nucleotides come together, AT, adenine binds with thymine, cytosine binds with guanine, catalyzed by DNA polymerase. These 2 strands are formed, one with the original strand, one with the new strand. They're exact copies, semi-conservative replication, okay? That means we will use 1 original strand and 1 new strand. Now we have 2 copies of all the DNA and ready for cell division are the mitotic phase, okay? There are 4 stages in cell division: prophase, metaphase, anaphase, telophase. They're easy. They are easy. 4 phases in the mitotic phase. Prophase. In prophase, the mitotic spindles form. The nuclear membrane breaks down. And the chromosomes, you know, in a normal cell, you can't see chromosomes. Okay, in a normal cell, you can see the nucleus, but you can't see the chromosomes. Well, that nuclear membrane is broken down. Nuclear membrane is broken down. Remember, we've replicated the DNA. Now we're going to have to divide the DNA and make 2 new nuclei. Okay. Nuclear membrane is broken down. The chromosomes are starting to be seen. Okay, they're starting to be seen. Okay, and spindle fibers, if you can see them, are starting to line up. So we go from a cell, a typical cell that has a nuclear membrane that has a nucleus, to the nuclear membrane being broken down. We obviously see formed components, which are chromosomes. They're in no particular order, okay? They're starting to line up, but they're in no particular order, okay? But we do see nuclear membranes broken down. We do see chromosomes. We do see form elements. And if we had high enough magnification, we'd see some spindles. That's prophase. Metaphase, they line up. They line up right here. Chromosomes migrate to the center of the cell, line up along the metaphase plate. This is metaphase right here. Remember, prophase is this. We see the chromosomes. They're starting to line up, but they're in no shape, no form, no fashion, or they're lined up. Okay. Metaphase, they're lined up. Their spindles are attached. Anaphase, they're starting to pull apart. Anaphase are starting to pull apart. Chromosomes are pulled apart. And in telophase, they have pulled completely apart and the cell is starting to divide. The cellular, so telophase actually refers to the nuclear component where the 2 separate nuclei, the chromosomes have pulled into what's going to be 2 separate nuclei. And cytokinesis is this part where actually the cytoplasm, the cell itself, is dividing. But as a general rule, everybody just calls this telophase, okay? So in summary, prophase, the nuclear membrane is broken down. You can start to see chromosomes. You can start to actually see chromosomes. And they're starting to line up, but they're not lined up. In metaphase, they're lined up along the metaphase plate. It's a straight line. If we were in the lab, we would look at onion root tips or any other plant that has a germinal root tip. Have slides from that, and you usually can see all the stages. You have to look for a bunch of slides, but you'll see a metaphase state, and it looks exactly like that. It looks exactly like that. Anaphase is when they're starting to pull apart. Metaphase, they were lined up. Metaphase, they were lined up. Prophase, they were not lined up. You can see the chromosomes, but they were not lined up. Metaphase are lined up. In anaphase, they're starting to pull apart. And in telophase, they have pulled apart. They're starting to reform a nucleus. To reform a nuclear membrane starting to form, okay? And the cytoplasm of the cell is starting to divide. And if you ever see this word cytokinesis, that means the portion of telophase when the cell actually divides. Remember, because mitosis, we're actually talking about, we're talking about the nucleus when we talk about stages, okay? But there again, most people just talk about telophase as the nuclei reforming, the 46 chromosomes in each one of the nuclei, and the cell divide. Those are the 4 stages. Cytokinesis specifically, technical definition.
I can't hear you.
I'm sorry, can you hear me now? Can you hear me now? Hello?
No, I can. No.
I can hear you now.
Okay.
There you go.
Okay, my internet connection is unstable. I live on a road which has a broadband cable. Okay, have a broadband cable. It has only one cable going down the road. Okay, the more people draw from the internet, the less service I get. So the one broadband cable is going right down the middle of our street. Okay, and everybody on each side of it, all the way to the 200 houses, are probably pulling from this one cable. So I don't know if you saw it, but it came up on my screen. I had internet interruption, okay? So mitosis is specifically division of the nucleus, whereas cytokinesis is division of the cell, okay? And so that's a technical definition, just so that you know it, in case, you know, somebody tries to trip you up. I wouldn't try to trip you up, but in case somebody tries to trip you up on a question or something like that. But frequently, when you talk about mitosis, you talk about both as telophase, okay? And so these are just the stages here. Stages, you know, prophase, metaphase, anaphase, telophase. It's not hard. Protein synthesis. Now this is a little hard. How do we make a protein? DNA codes for amino acids. That's the only thing DNA codes for, is amino acids. Every three base pairs in a row codes for one amino acid. So a triplet, so that's 3 base pairs. They could all be AAA, they could all be TTT, they could be ATC, they could be CAG, but they code. Now, the code may code, the 3 code may code for more than one amino acid. Okay, so you could have ATT and TTA both code for a amino acid. Okay. But it's a triplet, so it takes 3 base pairs of the DNA, AT, adenine, thymine, cytosine, and guanine. Those 3 in a row, in a linear row. This is the DNA's unravel, okay? In a linear row is a triplet, okay? They code for amino acids. Here's an example. CCT codes for glycine. Glycine's the only one I remember. Remember the amino acid structure: central carbon, a amino group, an NH2 group, a carboxy group, COOH, a single hydrogen, and an R group. Glycine, the R group is H. That's the only one I remember for. So CCT codes for glycine, AAA codes for phenylalanine. So if we've got to make a protein, okay, if we've got to make a protein, proteins are coded by way of DNA, by way of amino acids. DNA only codes for amino acids. That's all it codes for. Now, the protein may be modified outside of the nucleus, but all the DNA codes for is for amino acids, okay? Gene is a sequence of triplets that code for one protein. So 3 proteins is not going to be a gene. That's one triplet's not going to be a gene, okay? If a triplet is a word, then the sequence is a sentence. A small gene is 2,000 base pairs, okay? About 700 amino acids. If you divide 2,000 by 3, it's about 700, 600 something, okay? So the smallest gene is 2,000 base pairs, which is about 700 amino acids. Not all DNA codes for proteins. They are some useless segments, which are spacers. They were called introns. They are cut out. They're cut out later, probably in the Golgi apparatus. Remember, there's a lot of packaging and modification done in the Golgi apparatus. So there's segments, nonsensical segments, maybe stop signals, maybe start signals, maybe just spacing signals in the DNA code, built into the DNA code. There are also portions of the code that are known as exons, okay? The parts that are exon code for something. They code for an amino acid, okay? The parts of the exon, they're parts that code or something they're called exons. The nonsensical parts, which there again, I use that term, but it could be start and stop signs, or it could be just spacers. They're called introns. They do not code for an amino acid. Here's what happens. We need a protein. We need to get a blueprint out of the file cabinet. We need to take the blueprint to the factory floor, and based on that blueprint, we need to build something. Okay. The blueprint is the DNA. We need to copy that DNA. We got to leave the original blueprint in the nucleus. We got to copy the blueprint, send it out to the factory floor, read the blueprint, and assemble a car or whatever we got to assemble. In this case, we're going to assemble a protein. When we copy the blueprint, that is called transcription. That probably dates back to the stenographer that's transcribed what you were saying and then type the letter. Okay. When you code the DNA, okay, when you copy the blueprint, that's called transcription. When you send the blueprint out to the factory floor, they're going to translate that blueprint into a car, where they're going to look at that blueprint and say, hey, we're going to make a car by putting all these parts together based on this blueprint. So they're going to translate the blueprint into making a car. So in a nucleus, we have DNA codes only for amino acids. We need to build a protein. Protein is going to be modified. It may be changed into a steroid. We look at steroids, maybe changed into a steroid, maybe changed into a lipid. Okay. But we're going to start with a protein, because that's all the DNA codes for, is for amino acids. We've got the blueprint, the original blueprint. We're making a copy of the blueprint. We call that transcription. We send that copy out. We send that copy out by a messenger. RNA is our messenger. Okay. We call that mRNA, messenger RNA. mRNA. We take that blueprint out by way of a messenger. The messenger and the copy of the blueprint is mRNA. We then take that blueprint to our factory worker, which is the ribosomal RNA. We take that blueprint to the factory worker, which is the ribosomal RNA. The mRNA and the messenger RNA, and the ribosomal RNA are going to sit there, and that blueprint is going to be transcribed by the factory worker, okay, transcribed by the factory worker, in addition to our transfer RNA, who's the person who's going to transfer the parts in there to bring in the amino acid. So we start with our blueprint. We start with our DNA. The blueprint's going to be copied, okay? It's going to be copied by way of messenger RNA. So we're going to open up the DNA. We're going to copy the gene. If the gene is 2,000 base pairs, we're going to copy all 2,000 base pairs plus any start and stop signals they are, okay? So transcription is copying of the DNA and sending out that copy by way of messenger RNA. Translation is taking the messenger RNA, which is the blueprint, in association with the ribosomal RNA in the rough endoplasmic reticulum or in the smooth endoplasmic reticulum, and using transfer RNA to read that blueprint and bring in the amino acid we need, okay? Transfer of information from DNA-based sequence to RNA-based sequence. Ribosomal RNA are the components of ribosomes, and ribosomes aid in the translation process. So we make the ribosomal subunits in the nucleolus, go out the nuclear pore, go out into either the cytoplasm, or making proteins that are going to go outside the cell, are attached to the endoplasmic reticulum to make proteins. Ribosomes that aren't attached to endoplasmic reticulum make proteins that are going to stay inside the cell. Ribosomes that are attached to endoplasmic reticulum make proteins that are going to be associated with the cell membrane or go outside the cell. The example would be the thyroid hormone. That's going to be pushed outside the cell. So the ribosomal RNA, the subunits are made in the nucleolus, go out the nuclear pores, go out and be associated with either the rough endoplasmic reticulum or out in the cytoplasm. They're going to aid in the translation process. RNA translation process. Here's the subunits. Just need to know there's two subunits. This is a schematic here. This is our ribosomal RNA, okay? The ribosomal RNA on the DNA back here in the nucleus, we had a triplet. So if we had A, T, G, that was our triplet. That was on DNA. That's called a triplet. We then formed a codon on the mRNA. If we had an A on the DNA, we would have a U on RNA. We don't have T on U and on RNA. So we'd have an AU, a TA, a GC. So we have the exact same match, except in RNA, we don't have a thymine, so we have uracil replacing the thymine. RNA comes out as a single strand. That is our blueprint. So we started with a triplet in the DNA in the nucleus. We come out with 3 base pairs, which are called a codon. Those are the 3 base pairs that we coded for that amino that was on the DNA in the nucleus. They were transcribed onto the mRNA. So the triplet was transcribed onto the mRNA by way of a codon. The mRNA then came down to the ribosome and attached to the ribosome. It has this codon sitting here. We have a bunch of transfer RNA. These are transfer RNA. Okay, these are transfer RNA. This is an enzyme, which is going to put the specific amino acid together with this transfer RNA. There's different transfer RNA for the 20 different amino acids. These are out in the cytoplasm. They're going to be loaded up with amino acids just around in the cytoplasm, waiting for a blueprint to come out. When this particular codon is here, the transfer RNA has an anti-codon. In other words, it has the opposite of the codon. So if there's a U here, there's going to be an A there. So we have a triplet, a codon, an anti-codon. The triplet is on the DNA in the nucleus. Triplet is 3 base pairs. That transcribes onto messenger RNA as 3 base pairs, but now it's called a codon because they're the opposite of the base pairs that were on the DNA. So we have a codon here, the transfer RNA, which has the amino acid attached to it, which is why we're building the protein. We want to attach a bunch of amino acids. The transfer RNA then brings the anti-codon down to match the codon. By matching the codon, if you have a G down here, you're going to have a C up there in the middle. If you have a C on the end, you're going to have a G down here. You're going to have a U there. You're going to have a T there. Okay, that's why you got to know the relationship of what matches what. So this is going to be brought down here. This amino acid, this protein's being formed. We've got these amino acids being hooked up here and they're attached here. As soon as this transfer RNA unfurls its amino acid, it is released, just like this one right here is released here. This one's released, it no longer has amino acid. This next mention, there's a whole bunch of amino acids here with anti-codons. They're going to match up with this codon. They're going to bring in the next amino acid, and it's going to keep going until the messenger RNA is completely ended or until they get a stop signal. Messenger RNA is the messenger RNA. nucleotide strand that transcribes the DNA template into something that ribosomes can translate. That's what we're talking about, this messenger RNA. The ribosomes are here translating this and calling out the transfer RNA. I don't know how the signals could go. Okay, I don't know if anybody knows how the signals go. But the transfer RNA that matches this codon is coming in here with a specific amino acid. The transfer RNA, cloverleaf shaped molecule that brings the amino acid to the ribosome for the protein chain. So we're building up, so if we're looking at this another section, we came out of here, this was our DNA. DNA had a triplet. We transcribed that into a codon. This codon came out here and attached to a ribosome. We have a three nitrogen-based codon, okay? And these nitrogenous bases in our amino acid, in our sequence, this is a codon. This is the anticodon, which has a particular amino acid that was read by the ribosome, so that this transfer RNA, which is bringing an amino acid and transferring it to the protein, is going to come in here. And this is so you have a triplet, a codon, and anticodon. And then once this is released, and you have the next one in line, and you have another one here. This is an enzyme. Remember, enzymes have a lock and key formation where the transfer RNA and the amino acids are going to fit right in lock and key. They're going to undergo a configurational change, and then that amino acid is going to attach to the transfer RNA. Transcription factors bind to promoter site. DNA helix is unwound. mRNA strand begins to be synthesized, just like DNA replication, specific base pairing for DNA to RNA. In RNA, U takes the place of T. So I could envision you having a multiple choice question saying you start with this, give you about six or nine base pairs. This is what you start with: A, T, C, G, T, T, A, G. And what is your codon and what's your anticodon? Okay. And it's not a hard thing. All you do is just line them up. And this is DNA. So you know DNA has A, T, C, and G. So you have to be given that. RNA is going to match the A, T, C, and G, except instead of instead of the RNA having a T, it matches the U. That's why it's important to learn the facts that we learned in the macromolecule thing about nucleic acids. And so going to be U in place of the T. So we have a DNA template, the complementary template. So this would be the messenger RNA. We had an A on DNA. Okay. RNA doesn't have a T, so it's going to have a U. We have a T here, it's going to be A there. We have a G there, it's going to be C there. We have a C there, it's going to be G there. We have an A here, it's going to be U. We have a T there, it's going to be A. So this is our triplet. We have two triplets here. This is our codon. We got two codons. This codon is the antithesis of this, the matching of this complementary RNA. This is messenger RNA. The codon is the mRNA sequence that corresponds to the DNA triplet, leaves the nucleus and enters the cytoplasm. Sequences translate from codons to amino acid proteins. You never have to know this. When I took amino acid metabolism, they would give us a chart like this, and then they would give us the codons and things like that, and we'd have to translate into which amino acid is. But you don't have to worry about this. But this is how you figure out what amino acid you have based on these triplets. There is a ribosomal subunit, two segments attached. The tRNA has a dual recognition site, must have a code for the protein anticodon and anticodon. So the anticodon is the complementary base pair of the codon on the tRNA. So the tRNA has anticodon, messenger RNA has the codon, the DNA has the triplet. tRNA attaches to the appropriate amino acid. First amino acid is always methionine. Ribosome has room for three things: an incoming tRNA, an outgoing tRNA, and an mRNA strand. This elongates as you continue to add amino acids to it. Okay. They then attach. There is a termination. Okay. The process continues until a stop codon is reached. The ribosome then separates to start the beginning of the mRNA strand again. So if you need another protein, which you wouldn't just be making single proton. You also can have multiple ribosomes coding for a segment of mRNA. Okay, it won't be, you know, this, if they, you know, a gene, the simplest gene had 700 base pairs. Okay, had 700 triplets. Imagine if it had 3 or 4 or 5,000 triplets. Okay, you might want to incorporate more than one ribosome to code that section of mRNA. Okay, and this is just a sequence here. This goes through an illustration, okay, of what the DNA sequence was, what the mRNA sequence is. So this is triplets, this is the codon, this is the anticodon. This would be a similar thing that you might get on the question, on the short answer question. You know, have 6 base pairs, just 2 triplets. Well, and so you would have the triplets, ask you what the codons were, and ask you what the anticodons were. DNA triplet goes to an mRNA codon, goes to amino acid anticodon. DNA is transcribed into mRNA, which is translated to protein. There's that chart again. Cells can differentiate. We're the last 5 or 6 slides. Not all in DNA is used by each cell. I told you every cell is totipotent. The big toe cell has the potential to be a heart cell, but it has a signal making it a big toe cell. Muscle cells have a lot of actin and myosin. Thyroid cells produce thyroid hormone. Cells can send out signals to each other to turn genes on and off. You can have atrophy. If you've ever had a broken arm, had in a cast for 6 or 8 weeks, you took the cast off. You've had muscle wasting. If you've ever met anybody who's a paraplegic or has loss of nervous function in an extremity, the muscles there will undergo atrophy. Okay. You can have hypertrophy. If you work your muscles, they can build in size to a certain limit. You also have to have testosterone. So you can only get hypertrophy to certain reasons, limit. Hyperplasia is increasing the cell number or growth rate. So you can have hyperplasia. You might not want to have hyperplasia, but you can have hyperplasia. Metaplasia is one cell type placed by another cell type. That's usually not good, okay? You go from a normal cell to a metaplastic cell. So you might be talking about precancerous cell. You have pre-programmed cell death. Some cells are pre-programmed to die, either based on a signal or a time thing. That's apoptosis. You can have necrosis if you have a lack of blood supply. If you have peripheral vascular disease or diabetes, diabetes causes problems with the small vessels, and you have a lack of blood supply into the distal toes, you can have death of the tissue. Cell junctions, there's 3 types of cell junctions. Tight junctions, which if you look at, they are junctions between the plasma membranes. You have a tight junction between the plasma membranes, and so you have a junction here and a junction there with an open plasma membrane in between. So that walls off a space between 2 cells with an open space of interstitial fluid in between in which they can exchange material. So essentially we have washers at the top and washers at the bottom. It's a tight junction. We're trying to wall off a space, a potential space between these 2 cells where they can sort of control their environment, keeps things between cells and keeps other things out of the extracellular space. So we wall off the extracellular space between 2 cells, and we do that with tight junctions. A desmosome is a bolt, okay? As fibers, it goes from out one end of the apparatus into one cell and one to the other cell. This allows the cells to stay attached. You know, some tissues can elongate, okay? Elongate and then retract back. If we didn't have this anchoring system between the 2 cells, we potentially, you can rip them apart if you go past where the desmosome can act. But this is what keeps the cells together when they elongate, okay, is a desmosome. So we had a tight junction so that we could have a protected environment between 2 cells, okay? We have a desmosome which anchors one cell to the other. A desmosome anchors one cell to the other, allows for stretching and distribution of mechanical distress. And then we have a gap junction, which essentially is a tube between 2 cells, channel or tubes between cells that allows change of nutrients and ions. So you can actually have a channel with a gap junction, okay? You can have a tight junction that gives you a potential space between of extracellular fluid, or you can have a anchor between two cells to keep the cells staying together. Let me take this off. Let me open up the participants up here. Unmute everybody. Unmute. Okay. Any questions?
No, thanks.
No, thanks.
I think he's done.
I'm done. Yeah, I'm
Are you
done.
done?
So we're done. If you were not here at the first, I'm having to move the lectures to YouTube because I don't have enough storage space in my Zoom account. I have a paid account, but it uses storage space. So I'll be doing that over the weekend. I'll send you links to all the lectures. This will be the first lecture I put on YouTube, so I'll probably have it in the next couple of days. But I've got a YouTube channel. I just have to set it up to put on longer videos on the YouTube channel. And the lab module that was due next Monday is not going to be due next Monday because next Monday is a holiday. So it's going to be due on Tuesday. I have to change that due date if I haven't already done so. Okay. So that's all I got to say. We'll see you next Tuesday. Email me if you have problems. Bye.
Okay, thank you.
Thank you.
Thank you. Have a good night.
You too.
Professor,
Yes.
is the anticodon the same thing as tRNA?
No. tRNA is the structure, is that T-looking structure that carries an amino acid on one end and has the anticodon on the other end. So a tRNA, we have three types of RNA. We have messenger RNA, we have ribosomal RNA, and we have tRNA.
Mm-hmm.
Messenger RNA has 3 base pairs, which is called a codon. tRNA has 3 base pairs, which is called an anticodon. But the tRNA is just a structure that has an anticodon. It's the same. It always keeps the same anticodon, and it can only attach to one amino acid.
So the tRNA carries the anticodon.
tRNA has the anticodon on it. Yes, correct. And that
And
anticodon is for a specific amino acid, which it has an enzyme which it can fit into the slot, and it will attach to that one amino acid that is coded by that anticodon.
a conjunction of amino acids form an enzyme.
A
Or-
what?
Like a lot of
Amino acids can form an enzyme. That's one of the things it can form is an enzyme. It can form hormones. It can form literally every structure in the body. It just has to be modified. Normally, if you just have amino acids, they form proteins, and proteins are enzymes, right? The proteins are our structural things, okay? Proteins are, we have proteins in our body. So proteins have a number of functions.
so amino acids form enzymes that forms proteins.
No, amino acid forms a protein.
Okay.
And the proteins can be modified to be enzymes.
Okay.
Okay, so all DNA codes for is for amino acids. All you're going to build is an amino acid. Okay, all you're going to build is a protein. That protein can then be modified to whatever it needs to be modified to.
Mm-hmm. So DNAs or RNAs are proteins, correct?
They are, yeah, they would be proteins, right?
Okay.
They're also nucleic acids. Remember, a DNA consists of nucleic acid wrapped by histone proteins.
Okay.
So remember, we talked about the 4 macromolecules. We talked about carbohydrates, lipids, proteins, and nucleic acids. Okay.
Mm-hmm.
So there's 4 different macromolecules, but the only thing that DNA codes for is proteins. It doesn't code for the other things. They
Okay.
can be modified. They can be modified to make the other things, or they may be other cell organelles can make the other things. But DNA only codes for amino acids. But obviously, it has the code for everything in the cell.
Mm-hmm. I got it. Thank you.
You're welcome.
Bye.
Bye.